Good afternoon. This is Dr. Dan Guerra coming to you from VerEvMed Studios in the Pacific Northwest. Today I'm going to continue on in a series of lectures we've been moving towards that are all related to bioenergetics. Of course, the energy currency of the cell and energy metabolism in the cell. I've been focusing in on how energy metabolism is related to disease. And I've been doing this for the last, probably 20 or 30 different video lectures. Off and on, we've talked about fatty acid metabolism, carbohydrate metabolism, amino acid, nucleotide metabolism, various types of anabolic and catabolic pathways associated with the generation of ATP via usually the electron transport chain, which is embedded in the mitochondrium. Today, I'm going to focus in on how all of that comes together in a locus known as Alzheimer's disease, and in more general, neurodegenerative diseases. So let's get started. Okay, first of all, that's me, Dr. Dan Guerra, and I am the Chief Scientific Officer and co-owner, co-founder of VerEvMed. VerEvMed is a company that deals with looking at primary scientific literature as published in refereed journals in our trade, particularly in biomedicine, but also all extents thereof, including physiology, genetics, biochemistry, which is my specialty, pharmaceutical sciences, veterinary sciences, cell biology, and even areas that are only tangential to those, such as clinical pharmacology and indeed, of course, clinical medicine we also discuss. We do this with our clients on a one-on-one basis, with me as the primary lead on focusing on what they need to know from the scientific literature. I discover it, I extract from it. I look at the evidence that is published in a given area, a topic that someone's interested in, and then I present to them something like this lecture, but it would be a one-on-one Skype sort of interaction. And that interaction can lead also to written descriptors, as well as full-blown manuscripts, whatever is necessary for them to understand a given topic area and what the science actually says. Clients include people such as physicians, nurses, such people in the psychology industry, in the legal industry like lawyers, as well as institutions like hospitals. What we tried to do also, a major focus of VerEvMed, is to try to increase the number of people that we have from the lay audience, people who want to know more about a scientific aspect of, let's say, their disease onset, or let's say their pharmacotherapy, or what a recent diagnosis might mean to them, and what the science says. And when we do that, we work in conjunction and in league always with a physician, and then the physician in consultation with their patient, we discuss what the science says about a given topic that they're interested in terms of their health. We also cover nutrition, including nutraceuticals and over-the-counter therapies and remedies that are associated theoretically with a healthier life. We do quite a bit of that. So I've been a professor for about 30 years. I've taught general and advanced biochemistry in medical schools and in graduate schools. I've also taught medicinal chemistry, organic chemistry, general chemistry, cell biology, cell physiology, and of course genetics, and also straight pharmaceutical science. I've studied and taught research methods. I've published in all these areas, including most recently in the last, now 10 years, neuroscience. That's where most of my literature has been published in the last 8 to 10 years. So that's enough about me. Let's get started on this talk. I guess we need to say that that's my email address. If you want to contact me about the content of this video lecture, please do so. I will usually get back with you within 24 hours. This is my Facebook page down here. It has all of these recorded video lectures, as well as a lot of other things in philosophy and general things that I'm interested in, including some stuff about my family. And so let's get started. VerEvMed, of course, has its own website, and I'll show you the website and the email address directly to the VerEvMed website link. And when we do that, you can contact me through the VerEvMed as well. So today, the topic is going to be ceramide-mediated blockade of mitochondrial ATP export as in association with Alzheimer's disease. A paper published in the Journal of Lipid Research, JLR, just recently, late 2018. So let's get started. First of all, AD, Alzheimer's disease, is phenotypically characterized with the accumulation of something called amyloid beta peptide. So it's called A beta 42. The 42 has to do with its molecular mass. Elevation of that peptide and of sphingolipid of certain types, ceramide, which is a subclass of sphingolipid, and an associative mitochondrial damage, which can lead, of course, to programmed cell death. And if it's in the brain, we would call that neurodegeneration. Now, we know that culturing astrocytes, now I'll explain what an astrocyte is in a moment for those of you who don't know, with the A beta 42 purified peptide, what it does is it activates an enzyme called a neutral sphingomyelinase, isoform 2, which generates ceramide from sphingomyelin. So the substrate sphingomyelin, one of the products, is going to be ceramide, okay? So ceramide is something we want to key in on because it seems like it's linked to some of the pathophysiology and cell pathophysiology and biochemical physiology of Alzheimer's disease, particularly the early stages, prodromal, early onset, and it seems like perhaps even more so in female than male. Now, astrocytes, as I said I will tell you about, are a subclass of what we call white matter glial cells. Glial is a term that basically comes from where we get the word glue. And those glial cells in the central nervous system and the peripheral nervous system perform several key functions. Now, the glial non-neuronal, they're all non-neuronal, central nervous system cells. You know, the central nervous system is composed of neurons and then glia. What are the glia? They're astrocytes, oligodendrocytes, microglia, which I've talked a lot about in these lecture series, epididymal cells, and radioglia. The glial non-neuronal PNS, that's peripheral nervous system cells, include Schwann cells and satellite cells. And I've talked about those as well when I talk about neuromuscular junctions, for example, diseases associated with that, such as in sarcopenia associated with aging. All right, now let's take a look at this. I love these diagrams because they help you get a visual of what we're talking about. Of course, the colors here are just representative. They're not really what they look like inside the central nervous system. But you can see here that you've got a neuron, and it has its dendrites, and associated with neurons are microglia, which also have a lot of branching. You have astrocytes associated with axons that are myelinated, and the myelination is associated and derived from another glial cell called the oligodendrocyte. So you see how the neurons associated between their synapse from one neuron to another have embedded in this matrix of neuronal mass all of these glial cells, right? Oligodendrocytes, microglia, astrocytes are just some of them. So this here is a kind of a blow up of what a neuron looks like. I'm going to put on my fashionable glasses so they can read better. And basically what a neuron has is these dendritic spines, a cell body, also known as soma, which is Greek for body, a nucleus embedded in that cell body, and then an axon hillock, which is a constriction, and then this long axon. Okay, and the axon can be myelinated, and that is what we're really going to key in today. So a disease like Alzheimer's, Parkinson's, and even multiple sclerosis, and Huntington's chorea, for example, just a few of the diseases I can think about that are neurodegenerative, all are associated with demyelination of neurons in the central nervous system. And that myelin is actually a coating of lipid. So you get glial cells wrapping around the axon, and they do it in a punctuated down axonal radial symmetry that is punctuated by something called a node of Ranvier, which I've also talked about in these VerEvMed lectures. And that's basically increases that lack of myelin there, increases actually nerve transmission because it gives it kind of a jump or a leap between one myelinated region and the next myelinated region that enhances the speed of neurotransmission. So what these myelinated neurons do is they basically protect the neural signal, the action potential, as we call it, as it's moving down the axon, as it's getting from one neuron to the next, and that preparation of the neural transmission so that it goes down a myelinated axon allows it to be protected from any kind of stray voltage so that it moves quicker and cleaner, and the signal doesn't get disrupted or corrupted. So it's kind of like how we wrap insulation around a copper wire to prevent stray voltage. Very similar to the idea. So that's why I want you to understand the significance of what myelin is. So you have dendrites, that's these branching here, these spines, receive an electrical and chemical message from some adjacent neuron, upstream from it, okay, proximal to it. The cell body then processes that incoming information and takes the signaling, which is basically biochemical signaling, and generates an outgoing signal, usually the same signal, that's going to eventually generate neurotransmitters at the end of the axon. It's going to move down this axonal symmetry here. So the axon sends out the outgoing signals to the axonal terminals. And then the axon terminals make contact with the nearby, now distal to this initial neuron, new neuronal synapse, and then the information is flowed to the next neuron. That's how you get neurotransmission in a 1 minute sort of physiology. And this is, of course, a myelinated neuron. That's why I said it's really important. Myelinated, meaning it has a lot of these glial cells associated with this lipid layer, which insulates that axon, allows for rapid transmission. Here's more detail on that. So amongst the glial cells, you have microglia, astrocytes, and so-called tanocytes. And they've been most clearly implicated in metabolic control, actually, of the neuronal entire process. And this particular paper comes from Journal of Molecular Chronology. You can see the citation there. I'm not going to talk about tanocytes today. I'm going to key in on astrocytes. I'm also going to talk about microglia, which I've done a lot in the past. So I want you to see where astrocytes kind of fit in here. There's a neuron in association with an astrocyte, and also in association with oligodendrocytes. Okay, but these astrocytes are what we really are going to be keying in on. So what do they do? First of all, they're the most abundant glial cell in the brain. They involve neural support and communication. That's what they do. That's their function. They're also in a strategic geometrical position to sense nutritional status so that the astrocyte can send signals to the neuron to control bioenergetics, where we're getting the energy to carry out this neurotransmission. That's what we're talking about. What these cells actually do inside themselves. We know what they do as a functional unit, right? They transmit signaling, right? They transmit action potentials down that long axon, and so doing generate neurotransmitters at the synapse, and you get signal transduction cascades that lead to things like thought and ability to understand communication, like my voice, right? That's the outward whole global level of understanding of neurotransmission. We want to talk about what happens inside those cells. So they have to be able to determine nutritional status. Are they using carbohydrates or lipids, for example, for making ATP? And also what's going on with the blood, what's being brought in by the blood, what's passing that blood-brain barrier, which, by the way, the oligodendrocytes and the glial cells make up that blood-brain barrier. Okay. One of the things the astrocytes do is they oxidize fatty acids to generate ketone bodies, acetoacetate and beta-hydroxybutyrate, of course. And they also have lots of receptors, you might guess. They have to be able to receive information. They're going to have protein receptors embedded in the membrane. And that's going to be for metabolic hormones, okay? Steroid hormones, for example, adrenocortical hormones, even local hormones. There's sex steroids, of course, they're a whole part of that. They're corticosteroids. Cytokines, which are sometimes inflammatory, sometimes anti-inflammatory cytokines. Proteins generated by cells that carry out a response to stress or to damage or to ischemia, generating certain families of cytokines that can sometimes induce inflammation. That inflammatory response, if it goes unchecked, can lead to microglial activation. Microglial activation of the M1 type could, of course, turn into a global neurodegenerative process. And that's unfortunately what happens. And that's what microglia do. We're not going to talk about them today, like I said. So here's a paper from Pharmaceutical Research published about a year ago. So we're talking right now this is February 2nd, 2019. So that's about a year ago. So Alzheimer's disease, so let's do this general discussion now of Alzheimer's disease. We looked at some cell biology. I told you we're going to talk about ceramide, a lipid. I haven't scared you yet to show you what ceramide pathways look like. And I don't mean to talk down to you. If you're not afraid of pathways, then great, you're my kind of person. But we're going to do pathways, we're going to do structures, as we always do, because I am a biochemist, and I take no redress from that. I make no excuses for being a biochemist. I'm happy to be one. And so I like structures. Okay, back to clinical discussion. Alzheimer's disease, or AD, is the most common age-related neurodegenerative disease associated with incremental dementia. So it's absolutely dreadful. You're not only getting neurodegeneration, which means some of your faculties are declining, like, for example, your senses are declining, and eventually you die from this, but also you become less of the self than you were before. Now we call that dementia. So your cognitive ability declines, sometimes very rapidly. And this is just horrific. I'm fortunate that in my family we don't have Alzheimer's disease. I don't know anyone that's ever had it. We do have just age-related decline in consciousness and cognition, but we don't have Alzheimer's disease. And in general, that's not that uncommon not to have Alzheimer's disease. In fact, it's about 50-50. About 50% of the population seems to get it when they age. 50% don't seem to get it. So that's a curious aspect of the evidence and something we really don't understand yet. But I've talked about it in the past, and we'll talk about it again for sure, because research is always ongoing. Continuing, at the protein level, Alzheimer's disease is linked to both what are called neurofibrillary tangles associated with tau protein and the beta amyloid deposit, although a direct causal mechanistic link to those proteinopathies, because those are proteins and pathology thereof, that causal mechanistic link is not universal. So if you think we understand Alzheimer's disease, we understand a lot of it, but we also don't understand a lot of it. We know a lot of what's associated or represented by the disease, what it looks like, how it presents neurochemically and at the level of cell biology, and of course at the level of neuropsychiatric states. But we don't really know the true etiology of the disease and all the vagaries and varieties of Alzheimer's disease. All right, majority, 90%, 95% is idiopathic. It means we don't know where it comes from. So it's random. Major contributory factor simply just seems to be age. Every time you get to, every 5 years after 65, you can double the potential to contract Alzheimer's disease. Not a very good thing for if you have that age group. There is a rare congenital form, in fact, there are a couple of them, called familial Alzheimer's disease, or FAD, and it does have presumed genetic loci, but even there, it's a little bit blurry. We're not going to talk about FAD today, though. We're going to talk about the idiopathic, more common one. Since all Alzheimer's disease is characterized as the age-related progression of protein aggregate accumulation, there are these proteinopathies, it has a family recognition, quote unquote, with other types of diseases, neurodegenerative in sorting, and those are diseases like Parkinson's, or PD, and Huntington's chorea. Such proteinopathies obviously are implicated in pathoproteostasis. Okay, so they are linked in some way. That is, when I say proteostasis, that is the static level of these given proteins over time, which then would give you a time signature and denominator, which means it would be an event rather than a substance. Remember that I'm all about event ontologies in everything we discuss in nature, right? We don't talk about substances. We talk about things changing over time, and those are events, not just simple substances. You have to think that way to really understand biology, and I would argue to understand anything in nature. So pathoproteostasis can occur because of a corrupted protein turnover, because proteins do turn over, as with the ubiquitin proteasome system. It can be caused by excessive protein synthesis, for example, with the mTOR superactivity. Sometimes we see this in oncogenesis, or with an incompetent or incomplete autophagy. that is self-digestion of cells, which is a normal turnover. And often during starvation or stress, this is a very common preparatory mechanism so that you decrease the amount of mutations that can occur so that you don't get something like rampant cell division leading then to a tumor. Autophagy has been a topic of many of these lectures. And for example, you can take a look at one of the older ones there. I show you that link. So what happens here? Let's take a look at some more detail. Let's go back now to astrocytes. Astrocytes adhere to the central nervous system, responding to stress and damage. And they do that via a mechanism called reactive astrogliosis. That astrogliotic pathophysiology is emblematic of tissue and macromolecular lesions, damage, okay? Reactive astrogliosis is a spectrum of pathologies generating a continuum of context-dependent signaling as biochemical signaling events. Neuronal damage, for example, induced astrogliosis involves, for example, epigenetic reversible gene expression. So you alter the level of gene expression without altering the genome. So if you don't look at it in real time as an event, then you would never even know it occurred because it's freely reversible, leaves no mark. It writes an epigenetic code, it reads the epigenetic code, and then it erases the epigenetic code, all in the matter of milliseconds sometimes. That can alter gene expression and cell fate, leading to neurodegenerative disease, for example. Now, when you get epigenetic responses like that, you get cell proliferation and hypertrophy. That's part of this astrogliosis. And you also still maintain a preservation of cellular domains. That is, for example, microtubules and mitochondria. So we're going to talk a lot about this afternoon. You also maintain a reasonable amount of tissue structure. You're not talking about degeneration of the tissue. So that goes to long-lasting, eventually, scar formation with a rearrangement of tissue structure. And that, of course, can lead to serious lesions in the brain when that scar tissue is presented between, say, important series of nuclei, for example, for nerve transmission. So you have the physiological situation, the unchallenged central nervous system, and it carry out its physiological functions. Control of neuronal subsynapses, turnover, synaptogenesis, for example. Neurotransmitter uptake and recycling, such as serotonin reuptake. Maintenance of homeostasis, that includes not just neurotransmitters, but bioenergetics. What's going back and forth between the blood-brain barrier, very important. The regulation of blood flow, like how much, right? Vasoconstriction, vasodilation, we talked about recently. And of course, the control and modulation of new neurogenesis, right, to repair. So when you leave from that normal state and you get some kind of trauma, central nervous system trauma, and that can be an autoimmune disease, that could be an infection from an invading microbe, like a virus or a bacterium. It can also just lead, because of the neurodegeneration itself, which I told you is idiopathic. We don't really know what causes it. That all kicks in reactive astrogliosis. Now, in terms of the actual molecules involved, you get a lot of pro-inflammatory cytokines which turn this on. TGF-alpha, IL-6, NTF, as I said, interleukin-6, or even LIF. But a big thing we're going to talk about this afternoon is an increase in ceramide. That is this unusual sphingolipid we're going to talk a lot about. And you can take a look at that paper there, Brain Pathology 2012. It gives really good detail about ceramides involved in reactive astrogliosis. So now you can see that there's a lot more proliferation and hypertrophy of this cell type, right? So of these astrocytes. And that proliferation is in response to and helps handle, we believe, acute stress. It limits organ damage and tissue damage. Particularly, it's supposed to protect the neuron, because remember, this is an astrocyte. And eventually, because of its control over hemodynamics and bioenergetics, restore homeostasis. So reactive astrogliosis is disease-specific. So there are many permutations of this astrogliosis pathway. It can range from mild to very severe, leading a glial scar, which is definitely not good for the brain. It does show regional specificity. Some astrocytes turn on, some don't. And it can be diffuse or it can sequester a lesion area. Okay, it can spread or it can seal itself off. Persistent reactive astrogliosis can become maladaptive, and that's why it's been looked at as a potential target for attenuation as a therapeutic goal, again, when you're thinking about central nervous system pathologies like neurodegeneration. Now, we're going to start getting into a little bit of lipid metabolism. So now we're bringing in now another player. We already mentioned the name ceramide. Let's take a look where ceramide comes from. There are neutral and acidic sphingomyelinases. They've been shown to shuttle between the enzyme itself, between the plasma membrane and the Golgi. And that enzyme is actually activated by mitochondrial lipids. So lipids activate the enzyme. And what does that sphingomyelinase do? It takes sphingomyelin, which is part of that, remember the myelin sheath, and it degrades a little bit of it. It degrades it via this enzyme called sphingomyelinase, and it makes this lipid called ceramide. Okay? So when you look at the mole percent, ceramide is only about 1 mole percent compared to approximately 10 mole percent of sphingomyelin in cellular membranes. So the rest of it, of course, is phosphoglycerol containing lipids. Ceramide is made in the ER by another enzyme called ceramide synthase. Okay, so this isn't the only way you do this. You also can synthesize ceramide de novo, right? So that goes through this process here. This is the ceramide synthase pathway. Okay, so you can make it by degradation of membrane sphingomyelin. You can start off with serine and palmitoyl-CoA, the de novo pathway, and going through this palmitoyl transferase, and then dihydrosphingosine, dihydroceramide, and up to ceramide. Ceramide is a precursor to sphingomyelin, of course. All you need to do is add phosphatidylcholine. That's not it. Not only can you synthesize it that way, and that pathway involves adding fatty acid residues to long-chain bases like sphingosine and dihydrosphingosine. You can also hydrolyze the sphingomyelin that is catalyzed by different sphingomyelinases, distinct cellular compartments such as the plasma membrane, lysosomes, and Golgi. Now, not only that, you can also take you can also take sphingosine, okay, which is an intermediate in this pathway. And sphingosine can go through the ceraminidase to make ceramide. Likewise, sphingosine is in equilibrium with sphingosine 1-phosphate by this kinase reaction. And ultimately can finally be degraded this way, not to serine and palmitoyl-CoA, but to ethanolamine 1-phosphate and palmitylaldehyde. And this will not come back to the pathway. Now, there are places that these enzymatic steps leading to ceramide synthesis can be blocked by using drugs. One such drug we're going to talk about in our paper today is fumonisin B. Okay, so fumonisin B comes from a fungus called Fusarium, and it's actually a myco, that means fungus, mycotoxin generated from Fusarium. And this Fusarium is actually, many of these are molds. That's a wrong term actually, because they're actually fungi, but they are fungal diseases of plants. So this mycotoxin is actually generated from a Fusarium species that is known to affect corn and cause corn blight. Okay, so Fusarium is a very, very common form of this kind of disease. All right, and that's what Fusarium is derived. This fumonisin is a Fusarium-derived mycotoxin. That's what it looks like. Now, I'm showing you the structure because this branching and this long-chain alkane inundated with methyl hydroxyl groups looks a little bit like, not the same pathway, a little bit like certain sphingolipids. So it's believed that that particular drug, right, that mycotoxin, works by competing with natural substrates in these pathways. For example, it looks a little bit like sphingosine, and that might be why it blocks ceramide synthase here from sphingosine. Okay. So there's the ceramide synthase pathway, there's the de novo pathway, and there's the degradatory pathway. Okay. Now, stop here a second and think, ponder by looking at my woodpile. Neurodegeneration can be diverted, and it can be diverted via reprogramming of cell fate. By that, I simply mean what can the cell, any given cell, do? Let's say the astrocyte. It can be quiescent. That is, it might not change at all. It can be activated, as we saw in astrogliosis, which makes it proliferate, makes it get larger, turns on a lot of different metabolic machinery. It can also go through autophagy, start degrading itself, or indeed it can go through apoptosis and degrade entirely, destroy itself. Program cell death, right? So those are some of the fates that can occur. And neurodegeneration, one of the current therapies to understand Alzheimer's disease, maybe we can link into this neurodegenerative process and try to reprogram cell fate to prevent the process. That's the idea. All right, so let's take a look at this. It's somewhat of a complex slide, but it tells you all about Alzheimer's disease. There's a link for it. So you get oxidative stress, and oxidative stress is going to turn on this APP, amyloid precursor protein, really important for the proteinopathy of Alzheimer's disease, okay? Now, when that increases, a couple things can happen. It can be degraded, okay, by a secretase. So this enzyme here is a secretase. It's an enzyme. It's the beta secretase, okay? And that enzyme will cleave the amyloid precursor protein, APP, which is generated by oxidative stress, which is induced by transcriptional responses, including redox-sensitive transcription factors, okay? It's also turned on by translational machinery involving iron and this IRE, which is an iron-responsive element, transcriptional factors, which cause transcription translation of the APP. It kicks up. At the same time, oxidative stress turns on this beta secretase, and because it's a secretase, it can be secreted along with the precursor protein. And that is also reactive oxygen species sensitive, the transcription, and is translated through this PKR elongation IF2 factor, or eukaryotic initiation factor for translation. So that's how it's turned on. That's initiation factor, not elongation factor. Little e means eukaryotic. So you got beta secretase, you've got APP, you generate A beta 42. A beta 42 can turn on increases in free iron and copper, which enhances oxidative stress because of things like the Fenton reaction, generating reactive oxygen species, or the Haber-Weiss reaction, for example, that we talked about just last time. The A beta 42 also activates microglia. Microglia, the resident macrophages of the central nervous system, they can induce oxidative stress, including also the production of reactive lipid precursors, such as oxygenated fatty acids, like the prostanoids, and things like leukotrienes, but also microglia themselves generate pro-inflammatory cytokines. All that can enhance oxidative stress, rebuilding this pathway and increasing it and acting like a feed-forward mechanism, all because of the production of A beta 42. Likewise, you can also get an accumulation and interaction with the mitochondria, and that's what we're going to talk about today. You alter bioenergetics, you decrease it, and you increase the production of reactive oxygen. Now, we know why that happens because last time I told you that during the electron transport chain, if you block it in any way or corrupt it in any way, remember those four complexes, the fifth being the ATPase, proton pumping ATPase, you can generate partially reduced forms of molecular oxygen, and like the hydroxyl radical anion, and they can be potent oxidants to destroy things like mitochondria. They destroy nucleic acids, proteins, and lipids. So that's really bad news, right? So oxidative stress associated with increased expression of the APP and the secretase leads to the accumulation of the A beta 42. The clearance of A beta 42 from the brain is also blocked by oxidative stress. Okay, so you get more and more of it. Association with that with mitochondria and microglia and the production of metal ions lead to further oxidative stress, and it's a terrible, horrific cycle. We believe that this is one of the ways of looking at Alzheimer's disease. Okay, these pathways definitely are involved, right? And we're not sure that we can say universally or necessarily, but they're involved. They're intimately involved. We know that aspects of this are involved in these kinds of diseases. Now, I mentioned autophagy. Let me just bring it up again. I don't expect you to go back and look at. Autophagy can lead eventually to cell death, but usually it can control cell death. It can abate it. It can control cancer. It can interact with the innate immune response and also the secondary immune response. It can control or inhibit cardiomyopathy. It can slow down aging, the senescence process. It can work to diminish infectious diseases. It can work to diminish metabolic diseases like type 2 diabetes and fatty liver. And it also, the topic of today, autophagy, may be positive for controlling neurodegeneration. So basically, what does this involve? Well, autophagy, as it classically was understood, because phagy, of course, means phagos, to eat, right? So eating yourself, digest thyself. Always like to bring up a little bit of Shakespearean accent to these discussions when I'm lecturing. I don't know why. It's just weird. You don't need to pay attention to that part. Anyways, what you do with nutrient depletion or any kind of stress, you generate this AMP kinase, this AMP kinase. I've talked a lot about this in previous lectures. I'm not going to talk about it now. But imagine that you have ATP. ATP loses one phosphate, it's ADP, that diphosphate. Lose one more phosphate, you make adenosine monophosphate, AMP. That triggers a kinase, and that's what we're talking about here. So it's like energy depletion. You see how bioenergetics smuggles itself in. Right? At any rate, what that does, AMP kinase, one of the things it does is shuts off this anabolic system called TORC, that's target of rapamycin. You know, we talked about that in great detail too. I don't want to talk about it now. Basically, that's a generator of de novo protein synthesis and anabolic machinery, okay? Often a target for things like oncogenic tumors, right? Tumorigenesis. Anyway, when it blocks that, now look where these pathways work if you haven't seen these in a while. It blocks what mTOR normally does, and what mTOR normally does is block this aggregation of these proteins, leading to the aggregation of these proteins, some of which we've talked about a lot, like the ATG, and making that complex. When that happens, when that complex is made, because you're going down this place where you block that, so you de-repress this, so you allow this, okay? You make a phagophore, and a phagophore is on the way to making an autolysosome. So this is the autophagy process itself. All these other proteins, these ATG proteins, come in again with an LC3 protein. They isolate a membrane with cargo that is cytoplasmic cargo, so digesting what's inside the cell, that this phagosome is being created around, right? This autophagy being created around. You make an autophagosome. It can link with a lysosome, and lysosome has all the enzymes necessary to completely degrade what was enclosed inside that phagophoric complex. Basically what we're talking about here. These here are just showing you different inhibitors of these pathways where we've been able to study this. Now, resveratrol, which I've talked about, resveratrol, which remember, this is a phenolic compound found in grapes, for example, and cranberries. Resveratrol, an over-the-counter nutraceutical that people have been promoting. And I told you that it's a really important compound because it involves basically the acetylation and deacetylation of proteins, such as histones and metabolic enzymes, which leads to control over things, global control over things like initiating autophagy versus apoptosis, okay? And also the complete transmission of new gene expression. So resveratrol blocks it down here, whereas AMP kinase blocks it in a proximal way. So distal proximal. Resveratrol basically does the same thing, allows for this autophagy. All right. Normally growth factors and whatnot with insulin, this would be the anabolic mode, would turn on phosphoinositol 3 kinase. Can be blocked by any one of these drugs or compounds. Normally, that would turn on protein kinase D, okay? That kinase then would turn on the AKT, global kinase pathway, which triggers the mTOR pathway, okay? This is what happens, like, for example, insulin will induce this, and insulin promotes growth, right? Okay, so you get the idea. All right. So a dysfunction in autophagy, you see, results in poor clearance of that harmful amyloid beta or A beta 42 protein. Indeed, neuronal A beta contingent decreases with increasing autophagy. So the neuronal AB contingent, the accumulated concentration thereof, so I mean by the contingent, decreases with an increase in autophagy. So that's a good thing when it decreases, okay? In general, dysfunctional autophagy is observed in human central neurodegeneration. Okay, so that's why it's important to people studying neurodegeneration diseases. Functioning autophagy tracks pathogenesis of AD, where it serves to remit A beta protein and tau pathologies, both those proteinopathies, and functional autophagy does seem to be involved in controlling neuropsychiatric outcomes, such as in the prodromal phases, using some of these drugs, improve learning and memory, at least in the short term. None of these drugs consistently hold up. And I'll show you later how that works. It's pretty interesting. All right. Sorry about all this, but once again, let's get through this quickly. There are three types of autophagy. The chaperone-mediated autophagy, CMA, that degrades specific proteins containing a CMA motif, right? Some kind of like address. It's recognized by a chaperone complex. It's conducted to the lysosome where the protein is internalized to the LAMP2 translocation complex and degraded. Okay, so that's like a classical pathway, the CMA pathway. Here are all these compounds that inhibit this. By the way, notice how ApoE4 inhibits this whole process. That particular biomarker is linked to the potential for getting late onset traditional Alzheimer's disease. If you have a certain ApoE4 isoform of that protein, which actually comes from circulating high density lipoprotein passing the blood-brain barrier, don't you know? If you have a particular isoform of that, that can lock all this up and shut it off. We don't want to shut this off. So that's why that is sometimes a risk factor. If you have that allele, a certain mutant allele of ApoE4, it can enter through the blood-brain barrier via HDL uptake. And when that happens, it can block this whole thing. And that's not good because then you block autophagy. I just told you autophagy is important for a healthy brain. Right? All right. Now, there's also microautophagy. And you see this in Alzheimer's disease, Huntington's chorea, Parkinson's disease. These are proteins involved in it. And what they all do is block this pass. Okay, the disease itself, because it's neurodegenerative, is what pass is. It starts with a phagophore. Pass means phagophore assembly site. It just takes this phagophore, this isolated membrane associated with all those proteins we showed you, the ATG, BCL, etc. It closes up, it seals itself up, makes an autophagosome. It gets clipped, certain proteins get added, there's more ATG4 added to it, and ultimately it goes and associates with, remember, the lysosome, making a phagolysosome and carrying out this beautiful degeneration, which you want when you want autophagy to cook, right? So that's what PASS is. So if you have the disease, Alzheimer's disease, this pathway gets turned down, okay, because neurodegeneration has already started. And notice that what happens too is it can stop it later on. Like here is the phagophore trying to wrap itself around a mitochondria to get rid of it. So selective cargo for this pathway can be entire mitochondrial aggregates, entire endoplasmic reticulum, ribosomes, peroxisomes, pathogens, lipid droplets, you name it, whatever in there. Look out, we're hauling this in. This is again the macrophagy pathway here. Okay, so ultimately, then here are multiple places where we know the disease inhibits this late stream processing. So again, there's Huntington's disease, and Alzheimer's disease. A RAB5 protein is linked to blocking this. Parkinson's disease, the PINK1, the Parkin protein block this particular selective cargo uptake. You get the idea. Okay. So alterations in autophagy have been described for several neurodegenerative diseases, as you just see there. And the defects in autophagy machinery can occur in different steps along the pathway, and that's what I'm showing you here. That comes from a paper in Seminars in Cell and Development Biology, April 4 years ago. Boy, how time flies. Formal and efficient entry of pathophysiological sphingolipid metabolism. I love using some of Aristotelian causes, so I call it a formal and an efficient entry. If you know a little bit about Aristotelian causes, you know what I mean. You can look it up. Otherwise, it doesn't matter. You can just think about these as normal English words, formal and efficient entry of sphingolipid metabolism to become pathophysiological. Okay, let's go. Dihydroceramide, DHCER, or ceramide, just CER. Ceramide 1-phosphate, that's C1P, sphingosine-1-phosphate, S1P, and various gangliosides, which are sphingolipids associated with carbohydrate moieties, are all potent signaling molecules in their own right, and they contribute to pathogenesis of inflammatory diseases. They're also there naturally because they're necessary to carry out all the signaling and all the repair processes that happen during stress. So that's the other thing they do, right? So dihydroceramide is a precursor to ceramide. We just saw that in that pathway. In Denovo Sphingolipid synthesis via the action of an enzyme called dihydroceramide desaturase. There's two forms of them at least in humans, DAS1 and DAS2. DES1 is associated with electron transport that includes NADPH cytochrome B5 reductase. Remember that when we talked about the electron transport chain? Remember we're dealing with this NADPH process here? And cytochrome B5 itself, and it's active at the cytosolic phase of the ER. Okay, so this is the non-mitochondrial electron transport chain, such as in the ER and the peroxisome, because it's got the NADPH, and also the cytochrome B5 reductase that's actually associated directly just with ETC mitochondrial. Okay, this is all over the place, but you have to understand these organelles interact. Mitochondria, the ER, the Golgi, the plasma membrane, the nuclear envelope, they're always interacting. They're not just sitting there static like you see in a textbook in cell physiology. That's why I talk about event ontology. Event ontology, not substance ontology. You see? So what's wrong with everything? Taking a snapshot. You need a movie like this lecture. When DES1 is turned down or silenced, cells exhibit high levels of autophagy. That's interesting, okay? In part due to impaired ATP synthesis that leads to an activation of AMP kinase. Okay, so there's something going on here, right? So if you impair ATP synthesis, you jack up AMP kinase. I just showed you AMP kinase turns on the initial phases of blocking mTOR, allowing you to make those aggregates, allowing you to start that PHAGA4 complex, the PHAGA4 assembly system we looked at, the PAS system. So you see how this fits together so beautifully. So when you silence DES, what you're doing is you're not making ceramide, because you have a dihydroceramide desaturase, which is going to make ceramide, okay? And that desaturase is interesting because it puts in a, wait for it, a trans double bond in a fatty acid. I know you think the trans fatty acids are evil and wicked and so bad and so like non-natural. I am here to tell you that is total mythology. Trans fatty acids in biology are absolutely common and absolutely necessary. That makes them necessary and universal, which means they're part of the a priori network for cells to work. Yeah, smuggled in a little of Kantian metaphysics there, and of course, epistemology. All right, now, observation. Resveratrol, right, this phenolic compound you find in grapes, for example. You don't find enough find it in wine. I don't like alcohol, so I'm not going to say you find it in wine. You need to drink 50 gallons to get enough to have any effect. So you see what I'm talking about. You'd be dead long before you got enough resveratrol from red wine, from alcohol poisoning. So better to get it from another source like fresh fruit, I guess. There's another compound that works here, tocotrienols. Those are vitamin E components. And both of them may affect amyloidosis, which is, again, the A beta protein, in the animal model. So, and then we already know where resveratrol works. Tocotrienols work at about the same place, that PHAGA4 assembly process, and then shutting off that mTOR pathway. Resveratrol may induce autophagy after an increase in dihydroceramides, where it may inhibit dihydroceramide desaturase, which is going to make, remember, the ceramide. So you build up that hydroceramides, a precursor, and you don't block autophagy, you enhance it. You see, so that's why it's really important to understand, again, this complex event ontology, which ultimately is going to let me do in my last part of this series, discuss all of the things we put together in my new practical theory about biological systems, the diaeventontological theorem, which is going to put everything together. In order to understand, for example, Alzheimer's disease, we have to see it in motion. And we have to put together all the components and all the pathways so that we know what goes up and down and sometimes stays neutral or quiescent. And we have to know how they interact. So that's an event, not a substance. Making drugs to events may or may not work. I'm not saying they will, because I'm not a person who synthesizes pharmaceutical drugs, although I teach pharmacology I've taught it for several times. I'm not one that my mind isn't set on making these drugs, but you always hear about these, right? So I'm always worried about a substance like a drug affecting an event. In order to affect an event, you have to have an event, right? That's the way it works. That's another level. So keep that in mind eventually. All right, so let's go. So here is some beautiful, I know everybody was waiting for this, the Denovo Sphingolipid pathway. I know I dig this, and I know that the people that really love these video lectures also dig it the most, right? Sorry for saying the word dig. I know it's an old, like, beatnik term, and I'm not that old. My 8 years older than me cousin was barely a beatnik. Okay, so that gives you the idea. I was a little kid when beatniks were popular. Beat generation doesn't do much for me. And why am I talking about this? Let's talk about sphingolipid metabolism. Palmitoyl-CoA, which is just a thioester of palmitic acid, the product of de novo fatty acid synthesis and also degradation of triacylglycerols. Palmitic acid, as a CoA thioester, reacts with the amino acid L-serine, and using pyridoxal phosphate, which we talked recently, right, the PLP-mediated enzymes, is going to carry out this reaction in the presence of manganese at the 2+ form, making 3-keto-dihydrosphingasine, release carbon dioxide, which drives the reaction, and release free coenzyme A. Sorry, I was looking at my dog, make sure it wasn't doing anything bad. Okay. Now we're going to take 3-keto-dihydrosphingasine, and we're going to reduce it, and we're going to make sphinganine, and that is actually dihydrosphinganine because of this level of unsaturation here. Continue on. All right, so now we've made sphinganine. Now there's ceramide synthase, okay? There's ceramide synthase, which is going to add a fatty acid to this nitrogen, to this amino group, which was originally the alpha amino group from serine, right? So now you've made this N-linked fatty acid amide. That's dihydroceramide. Now this enzyme we just talked about, dihydroceramide desaturase, again, you're using NADPH, and look what you've done. You've introduced a double bond. I put this little circle around there just so you can see it. And that double bond is a trans double bond, a trans double bond. This is a trans fatty acid. So trans fatty acids are common as all get out in biology. You don't make any of these really important myelinating lipids unless you have trans fatty acids. So how important is that? Again, absolutely and necessarily. Okay. A priori requirement, right? Apodictic, if you want to call it that. All right. Now, you can also make ceramide via this pathway from sphingosine itself, right? There's a ceraminidase which can cleave off that fatty acid. Okay. Multiple ways, we already looked at that. You can also make ceramide from complex sphingolipids, such as sphingomyelin and glycosyl sphingolipids, right? Right, the cerebrosides and the gangliosides, for example. And we already talked about this full degradation pathway via this lyase, making hexadecinal, that's the aldehyde, and ethanolamine phosphate, which can be reused to retail lipids, of course. All right. Now, here's a little thing I told you I was going to bring up later on. Here we are. The antitumor drug doxorubicin, shown up here, entering inside a cell. Now, what doxorubicin can do to initiate the death of a tumor is it can turn on ceramide synthase and sphingomyelinase, thus triggering ceramide production, thus causing apoptosis. If it's a tumor cell, we're done. Now, doxorubicin actually works directly on DNA and RNA, but that damaging of DNA and RNA turns on, of course, stress responses such as the synthesis of ceramide. See how this works? Downstream event ontology. Now, the problem is constant dosing of this chemotherapeutic doxorubicin. Look what it does. It builds up ceramide, builds up ceramide. Finally, DOX-mediated glucosyl ceramide synthesis occurs, and there's a transcriptional response. It turns on this enzyme by working on this transcription factor XP1. You make the GCS, you induce the GCS gene, that is, you induce transcription. You make the GCS, that's the glucosyl ceramide synthase enzyme, and rather than ceramide generating apoptosis, which we already said we don't like in terms of neurodegeneration, this is now the central nervous system in its normal state worried about Alzheimer's disease. This is a tumor, it could be in the brain though, however, think about that. Buildup of ceramide, you can cause actually neurodegeneration if it happens there. Not good. That's why chemotherapeutics are very toxic. But look what happens. After a while, you get resistance to the drug because you induce this gene, which makes this glycosylated ceramide, which is inert and can't go back to make ceramide and doesn't induce apoptosis. You see what I mean about event ontologies. You see what I mean about how everything is integrated in a dialectical event ontological paradigm. That's dia-event ontology. And that's what I'm going to talk about at the end of this series of lectures. And it's my new paradigm. It's something I'm working on right now. There's going to be a chapter in the book I'm writing. All right. Now let's go back to this farm research paper for a minute. It examined the effect of inhibiting this desaturase enzyme in primary neurons from a double transgenic mouse model expressing human mutations in the APP and presenilin. That's another protein involved in Alzheimer's disease and Parkinson's. The neurons of these transgenic mice accumulate A beta as diffuse neuritic plaque in an age-dependent manner, and they display some dysfunctional neuronal proteins. So what is this? Basically, it's a mouse model for Alzheimer's disease. DES1 inhibitors, XM461 and 2, induce autophagy. Told you that's good here. In both wild type and transgenic neurons. Okay. So even the neurons that have this enhanced production of APP and presenilin, you see. This induced autophagy is associated with changes in neuronal lipid profiles. Why wouldn't it, right? Reduction of ceramide happens, certain sphingomyelin species, and glucosylceramide, and concomitant increases in dihydroceramide and dihydrosphingomyelin, and those seem to be good markers for autophagy. Those are biomarkers for autophagy in situ. Maybe not so good in serum, but people are working on that. Consequently, there is a significant reduction in amyloid secretion in neurons isolated from transgenic APP-PS1 mice, and that's all good. So this is an animal model knocking out the desaturase enzyme to make ceramide. You get all these other lipids building up because you don't have ceramide produced, and that seems to be affecting amyloid activity, and that also downstream may be a good target, right, for pharmacotherapy in human. All right. Here's a recent report published a year ago now, not just published. January of 2018 is done, so February 2019. Wow, again, does time fly? First published again in JLR, a journal I absolutely love. I actually think I like JLR better than JBC. There it is, I said it. This paper was inhibition of acid sphingomyelinase. Okay, look what it makes. Boom. Oh, wow, look at that. Disrupts lysosomal nutrient signaling complex, the LINUS, and triggers autophagy. See, back into lysosomal nutrient signaling. This is another component of that phagophore complex. We're just checking out. And that paper supports a PharmSci manuscript since a decrease in ceramide from an alternate pathway, it's an alternate pathway, not the desaturase pathway. Yep. Indeed enhances autophagy, presumably by blockading apoptosis. All right. Now, I said I didn't like JBC as much as JLR. I like JBC just fine. Here's a paper from a few years ago. Now, let's bring in Sirtuin. Sirtuin 3 mediated deacetylation. Sirtuin is a deacetylase. It removes acetate from proteins, histones, and also proteins and enzymes and cytoskeletal proteins, you name it. So SIR3 mediated deacetylation of the enzyme ceramide synthase increase the enzyme activity and therefore increase ceramide accumulation. So here you got SIR3, which is, a lot of these acetylases are being looked at as good targets to enhance the activity of, because when you deacetylate histones, for example, you decrease overall gene expression. And if you do that in a tumor, what happens? A tumor can't make all this necessary proteins, and it dies because of what? Autophagy, senescence, or even apoptosis. So SIRT3 is sometimes looked at as a good thing. In fact, remember the resveratrol? This is going to be involved in this process. So this pathway could be enhanced if SIRT3 is specifically activated by resveratrol. Resveratrol activates the deacetylase. Paper in PharmZ, 2016 paper, citation there, demonstrated indeed SIRT3 is activated by resveratrol. So ceramide synthase could be activated because you're deacetylating ceramide synthase. And I just told you ceramide is bad news for neurodegeneration. Right? Good news for cancer, but bad news for neurodegeneration. So this, you see what's happening. So resveratrol, all those 50 gallons of wine that I guess somebody would drink before they died of alcohol poisoning, or eating 3, 4, 5 pounds of grapes a day would probably give you enough of this stuff. Again, not something that would be considered very practical. Resveratrol that you could go into a pharmacy right now down here in Lewiston, Idaho, and buy some right off the shelf. Look what it's doing. So here is one of these nutraceuticals, right? One of these alternate medicines, right? That are touted by some people. Oh, this is the greatest thing since sliced bread or milk or whatever, water, I would say, or actually coffee. That's the best. That's one of the best inventions ever. Caffeinated coffee in particular. Um, Look, this may not be what is what you want, because it could also enhance ceramide synthesis in the brain. And if that happened, it could be linked to that whole what? The neurodegenerative process, because ceramide production. So watch out when you start taking over-the-counter alternate medicines. Now, another critical point before we get to the final paper. I'm sorry to take us so long, but let's go. Sphingolipids play a role in lipid raft formation, not draft. And these rafts, these are just flotillas of lipids and proteins, and nucleic acids and carbohydrates, transport the amyloid precursor protein and the beta secretase. So the actual production of A-beta-42 is directly linked to lipid composition of the raft. Huh. It surprised me, because sphingolipids, right? They're molecules that are amphipathic. They're working with the cell. They're dealing with, cores of hydration and dehydration. Because of hydrophobicity, they're going to move in the cell. Non-randomly, by the way, because of the structure of the lipids and the aqueous solvent, salt concentration, things like that, pH. Therefore, the inhibition of desaturase 1 or activation of ceramide synthase might reduce beta secretase activity, or reduce the membrane domains containing APP and beta secretase, impeding amyloidogenic processing of APP. Now, here we're showing you another alternative. We're saying that if you inhibit desaturase, right, or inhibit ceramide synthase, you can reduce the beta secretase, okay? You can reduce the beta secretase and or reduce the membrane domains containing the amyloid precursor protein because of this role up here, right? And in so doing, right, impede amyloidogenic processing, and therefore maybe another target for Alzheimer's disease pharmacotherapy. Furthermore, this pharmacotherapy might enhance the autophagic flux, as we just saw, because you're getting rid of ceramide, which can lead to the generation of less A-beta peptide anyway from the pathways, the canonical pathways I just showed you. So therefore, desaturase 1 might be a novel pharmacological target. So those inhibitors could one day be something, and maybe they're already out in phase 2 trial. I'm certain they are in some locations. Now, an increase in ceramide levels promotes apoptosis. I hope you're convinced of that now. The distribution of ceramide in rafts, or more extended ceramide-enriched compartments called CECs, provide multiple signaling paradigms. So once you put something in a raft, you can do all kinds of different signaling, right? Because you're moving around. An exclusive antibody to ceramide, that lipid, applicable to super resolution microscopy using stochastic optical reconstruction microscopy, or STORM. So this is a stochastic, right? What does that mean? That means a random optical reconstruction microscopy. It's stochastic random because it resolves around the way that this microscopy works where the antibody is. It kicks up the focus because it's inducing a signal, because the antibody is going to be linked to a fluorophore or to some compound that's going to induce light signaling. Isn't that cool? That's what STORM does. So that's all done. The people that published this paper we're going to be talking about generated the STORM system to help identify tubulin binding to the lipid. So ceramide actually binds to a protein called tubulin, which is, of course, associated with microtubules, which is part of the cytoskeleton, which I'll get to right here. Cytoskeletal tubulin polymerizes to form microtubules. Okay, that's what's going on here, where it binds 2 moles of GTP, one in an exchangeable site in the beta chain and one in the non-exchangeable site in the alpha chain, putting together these microtubules. Okay? And you can find all of that at that link. This is also found in Trends in Cell Biology, and that's where I got this particular see it and understand it diagram. Okay? So this literature is from that link. This slide, this picture is from that paper. So microtubule targeting tubulin binding compounds like taxanes and vinca alkaloids are established anti-cancer drugs. For example, a really nasty one known as vinblastine corrupts microtubule formation. Now, why do you want to corrupt microtubule formation? Because that's a spindle fiber involved in what? Cell division, right? Microtubule cell division, right? Mitosis. Yeah. I know you guys all remember this stuff, especially you doctors. So these certain pharmacotherapeutic, chemotherapeutic drugs like taxanes and vinca alkaloids, they block this stuff, right? And now I'm telling you, ceramide binds to tubulin, and tubulin is the monomeric protein making up this filamentous column of proteins, eventually turning into the microtubule. So ceramide back again in play, discussing microtubules. Now, why does this matter? Because the drugs used to control the tubulin ceramide bonding, right, could be redeployed for Alzheimer's disease. Yeah, you see that? That's what pharmaceutical drugs are always looking for, by the way. Anyways, this is all discussed in this PNAS paper in 2014, of course. Tubulin can be post-translationally modified, by the way. Oh, yes, it can. It can be tyrosine-elated. In other words, you can add tyrosine. You can remove tyrosine. You can acetylate it. Look out also for deacetylation, right? Like the Sirtuins. It can be phosphorylated. It can be palmitoylated. It can be glutaminated. And it actually can be glycated and sugars added. All that comes from my paper in Methods of Cell Biology, which I looked at. I wanted to know that because I want to know everything that's happening to tubulin, because I'm generating, you see, a diaeventontological discussion here. So everything has to be brought in because eventually we're going to put it all together and explain it. So ceramide can bind to free tubulin in the cytosol or microtubulin bound in that complex we just looked at, that columeric filamentous protein, right, called the microtubule. Or it can be associated directly with cellular membranes. Wow. Membrane tubulin is in the plasma membrane, so it's there too, not just in those microtubules. It's also in the endoplasmic reticulum, mitochondria. It's all over the place, okay? So tubulin specifically binds to lipids in the cytosolic leaflet of the outer mitochondrial membrane, or the OMM, to initiate closure, whoa, wait for this, a voltage-gated dependent anion channel called VDAC1. So it's going to initiate closure when tubulin binds to that. That VDAC mediates that co-transport I talked about last time, where ADP comes into the mitochondria and ATP leaves the mitochondria. So you can see if tubulin binds there, it blocks the VDAC, ATP builds up inside the mitochondria. That, as it turns out, dear listeners, is related to one of the pathological characteristics of the neurodegenerative disease known as Alzheimer's. Okay. My dog Rocky here looks like he wants to go out. I'm going to get up real quick and let him out. I'll be back momentarily. It's actually not my dog, it's my son's dog, but he's not here today. Okay. So tubulin specifically binds to lipids, as I just said, and it involves then this whole thing of shutting off ATP transport. You shut off ATP transport, you don't get ATP in the cytosol, you can't use it, right? So that's the point there. Now, sorry all the writing, but let's get through it. In Alzheimer's disease, A beta has been shown to bind to VDAC. Okay. A beta binds to it as well. That reduces ATP levels and causes mitochondrial fragmentation in prodromal and early stage Alzheimer's disease as a biochemical pathology leading to neurodegeneration. Oh my gosh, what about that? A beta does too. Tubulin and A beta. Who would guess? In astrocytes, VDAC1 is critical for the secretion of ATP. So astrocytes, remember, they're involved in bioenergetics. They secrete ATP. Okay, that's one of the things they do, which functions, sorry, there's an O in functions, as a gliotransmitter. Right, gliotransmitters. In other words, ATP is leaving the astrocytes, and now it's a gliotransmitter. Love all these terms. Regulating nothing at all except maybe really important synaptic plasticity and neuronal function. See that? So ATP doesn't just act as an energy source, a bioenergetic compound. It also functions as a transmitter. We know that from lots of other things, such as sleep research. Now, VDAC1 closure by tubulin is associated with a tumor-linked Warburg effect, which is, of course, we talked about also many times here. It's a metabolic switch from oxidative phosphorylation to aerobic glycolysis. So glycolysis is the presence of oxygen for ATP production. It's pretty wimpy for ATP production. That's what the Warburg effect is. So the lipid-tubulin interaction is important for tubulin-mediated VDAC closure. Now, the reason that Warburg effect occurs is because you can't get ATP out of the mitochondria. all you got is glycolysis. You got to kick it up, and that's why you get aerobic glycolysis. That happens in tumors all the time, by the way. So you close VDAC, you turn on tumor glycolysis, which is really a very wasteful way of generating ATP and burning off of the glucose, which is involved in wasting, actually. So the lipid-tubulin interaction is important for tubulin-mediated VDAC closure. We said this. Ceramide is enriched in mitochondria. Yep. And in mitochondrial-associated membranes called MAMs. MAMs are basically a subcompartment, how about this, of the ER. So mitochondrial-associated membranes can actually be a subcompartment of the ER. These membranes move around just like other biomolecules, membrane-associated clusters, okay? They don't cloister, they cluster and aggregate and move. Just thought that, I like that. They form contact sites with the mitochondria, and they take part in crosstalk between the mitochondria and the microtubule in association with the endoplasmic reticulum. See, things are really happening all the time in the cell. All right. Recall that ceramide synthase and the neutral sphingomyelinases generate ceramide in the mitochondria, and they're involved in inducing apoptosis, right? But it hasn't been demonstrated that ceramide regulates the function of these MAMs. Does it? We don't know. And whether this involves ceramide-associated tubulin, which they call CAT. Sorry about that. See all these acronyms? You know, you have to have almost a disinterested despair over these acronyms. Disinterested means I don't really care, I'm impartial, it doesn't really have to do with me, because the despair I feel with all the acronyms, I think everybody feels. You get way too many acronyms, it gets way too complicated to remember what they mean, right? I say it's disinterested despair, though, and I know that seems like an oxymoron, but you see how I figured it out? It's not. At any rate, there is a hypothetical deduction here with all this silly rhetoric I'm spewing, although it's not silly, it's not rhetorical, and I'm not even spewing, I'm just talking. The hypothetical deduction that's rendered in the central nervous system in situ ceramide production is closely associated with ATP depleting VDAC closure linked Alzheimer's disease, neurodegeneration. I put that all together in one sentence. I'm very proud of my hypothetical deduction. Okay, that's where we're going to lead to now. Now, we're in the paper. Experimental dialectics, get after it. If ceramide-associated tubulin does control or regulate mitochondrial event ontology, then it's occurring at the mitochondrial membrane locus. Okay, I think we can say that. Okay, proposition. The deployment of 2X knockout mice derived neutral sphingomyelinase knockout astrocytes and ceramide synthase, chemical inhibition via that fumonsin, remember that mycotoxin, as tools to discover morphological and microscopic mitochondrial alterations may not provide evidence, right? So thesis, antithesis, may not provide evidence in favor of CAT, remember ceramide-associated tubulin, on the MAM, remember that is mitochondrial associated membranes, mediated gain of function. So that's the what? The disquieting what? Antithesis. Now we're going to do the synthesis. That's why it's a dialectic, you see? The synthesis is actually that ceramide-associated tubulin localized at ceramide-enriched mitochondrial-associated membranes. So these are now going to be called CMAMs, ceramide-enriched mitochondrial-associated membranes. Sorry. Do generate an interaction, synthetic process here, between tubulin and VDAC, thus suggesting there is a ceramide-associated tubulin, and it does control or regulate mitochondrial event ontology, then it is occurring at the mitochondrial membrane locus. Okay, so see all of that? Thesis, antithesis, synthesis. And this is what we're going to key in on. This is what this paper is going to look at. It looks like this is true. Using their STORM technology, remember that stochastic optical reconstruction microscopy, which focuses in when something lights up, a ceramide-specific antibody binds to and visualizes ceramide in the cells and tissues. That's so cool. Got an antibody to a lipid. I think that's cool. It's a lipid biochemist. We always said we needed those. Here they are. This research lab, great for this purpose. That's why they're allowed to publish in the all-great journal JLR. Also developed an imaging protocol involving cross-linking a photoactivatable ceramide analog. It's a bifunctional ceramide called N9,3-pentaphorenyl 3H diazarine 3-eel nonanoyl d-erythrosphingosine. Nice name. I like it. I don't know that I'd be going around talking about it all the time just like that in any given conversation. Maybe I would with my son. Maybe. But let's just call that pack fasser, right? Or pass fasser. Let's call it pack facker. That's bad. Pack fasser. Okay. And you can use fluorifies or biotin to catalyze that photovoltaic event so that storm is turned on. Hope you're following. That's so cool. Reminders before the data. Sorry, almost there. The A-beta 42 leads to increased levels of ceramide. Told you that. In primary cultured astrocytes, and in neutral sphingomyelinase 2 deficiency reduces, so when you get deficiency in that enzyme and you reduce ceramide in the system and sensitivity of astrocytes, you get less A-beta induced apoptosis. Okay, that's the key here. So A-beta 42 induces fragmentation of mitochondria. I want you to remember that. And so the first data examines if mitochondria in sphingomyelinase deficient astrocytes were resistant to A-beta induced fragmentation. So here we go. Data. Figure 1, I believe it was. Yeah. Figure 1A shows that overnight exposure to a micromolar A-beta 42 led to fragment of mitochondria in wild type, but not in sphingomyelinase deficient. Okay. So you did not get a fragmented mitochondria, which is what you're seeing here. When you blow that up, it's fragmented mitochondria. You don't get that when you don't have ceramide production. Okay, so wild type and sphingomyelinase deficient astrocytes were incubated overnight with a micromole of A-beta 140 of that peptide. Mitochondrial morphology recorded after labeling with a mitotractor. That mitotractor is pseudocolored in red. That looks at mitochondria. Okay, and the panels on the right side are shown. There's the scale at 20 micron versus 2 micron. Just a blow up is all that is. Now, check it out. Fragmentation led to about a 50% reduction in mitochondrial length, aspect ratio, and form factor. So when you have, okay, the control, and you add the A-beta protein, right, it's the control system. The wild type astrocytes, you get what? You get less mitochondrial length. That's not good. That means you're getting a fragmentation. You're getting less of the aspect ratio, okay, when you hit it with the A-beta protein. Notice when you have no ceramide production from the sphingomyelin because you knocked it out, no effect. You have the A-beta protein, you do not get decreased length, decreased aspect ratio, decreased form factor. It stays the same. So removing the source of ceramide doesn't give you this fragmentation of the mitochondria, thus requiring you accept that synthesis from that last dialectic. Okay? Now, let's continue. This was a movie. I'm not going to play the movie for you, but if you look at the paper, you see the movie. It's kind of cool. Time-lapse images of the mitochondria, wherein the sphingomyelinase deficient, or now we're using that fumonisin B1 treated astrocytes, were rapidly moving. Okay? While those in the wild type, so these are moving around, while those in the wild type weren't. They were sitting down. They were resting. They were taking a snooze, okay? Indicating an unknown function of ceramide, the regulation of mitochondrial distribution and or motility. So it looks like when you get rid of ceramide, the mitochondria move around. They swim around inside the cytoplasm. That's a good thing. Why? Because they're interacting. They're carrying information. They're carrying around ATP, which will allow them to induce, for example, have enough metabolic machinery to induce autophagy. Oh. Ceramide stops that stuff. So that's all this says. Just, shows you that, the different coloring. You have the cytopainter pseudocolored in green. The T1, which is a component of this system, is pseudocolored in red. Motility of the perinuclear and peripheral is change in the deficient. Okay, so in the perinuclear, there's no change. But when you look about what's going on with the mitochondria moving around in the periphery, right? In the periphery, the mitochondria are moving like crazy. That means that ceramide removal allows a mitochondrial movement, which is an aspect of delivering ATP, which is a necessary component for autophagy to turn on. Second slide. Oh, this whole JLR paper had these beautiful colored slides. This is all the storm and all this confocal microscopy. Ceramide is co-localized with these membrane-associated mitochondria-associated membranes, peripheral mitochondria, and microtubules, which is all prevented by sphingomyelin deficiency. Okay, and of course, knocking out ceramide synthesis. The two different ways of synthesizing ceramide: ceramide synthesis or the sphingomyelin. So immunocytochemistry, that's what this is here. Beautiful, right? Using the mitotractor in blue. Okay, it's a blow up of that. The rabbit antibodies against ceramide, that's red. You can see that. And the MAM specific, right? The mitochondrial associated membrane specific sigma R, that's the green, all there in the mitotracker, right? So the left panel shows the overview. This shows you how they track in distance. It's an event. You see it moves, right? Showing you where you get all three coming together. You get the mitochondria, the ceramide, and the membrane together, okay? And you turned it on by this fluorescence signaling intensity along the dashed line that you're looking at here, right? And that leads to, and that's the white color in the image overall, this dashed line. The y-axis in the middle panel shows the arbitrary units of fluorescence intensity. So you increase, okay? Now, take a look at this. Co-localization of ceramide, that's red, with mitotracker, that's the mitochondria, that's green, in the wild type, and the sphingomyelin is deficient. And look what you see here. You see here that without that ceramide, you get what? You get the lack of co-localization of ceramide with the mitochondria. And we already know that when you co-localize the ceramide with the mitochondria, it stops moving, which we know is associated upstream from inhibiting the phagocytosis pathway, right? The autophagy. So when you get this dump here, this decrease, when you get rid of ceramide synthesis, this is good. Okay, and that's what you see here. Nice mitochondria, not associated in this now fixed gel matrix. Okay. Likewise, co-localization of ceramide shown here in red again, and acetylated tubulin. Remember, tubulin can be altered covalently post translational covalent modification. I told you that. You add the tyrosine, remove the tyrosine, acetylated, ubiquitinated, phosphorylated. This is acetylated tubulin, shown in the blue, in wild type of sphingomyelinase. The mitochondria are pseudocolored green. So this is all good stuff, okay? You don't have ceramide associated with the acetylated tubulin either. And that looks like it's linked to the mitochondrial movement, and that looks like that's linked to whether or not you get the pathophysiology, right? All of that is removed from getting rid of a source of ceramide. Ceramide forms contact structures with microtubules and co-distributes with tubulin. We already know that. So it's a confocal immunofluorescence series here using the anti-ceramide rabbit antibody, which is pseudocolored in red. The anti-acetylated tubulin mouse IgG, that's green, so that's the acetylated tubulin. And now you're putting it together, and that's a so-called Z-scan projection onto one plane. Okay, you see that they co-localize. Now, B, there's ceramide, there's acetylated tubulin. You're just blowing this stuff up is all you're doing. You're blowing up this pictograph here, this micrograph, right? And here's your overlay, and there's your storm. So what this is showing you is that storm using antibodies described. Remember, we got antibodies to ceramide, antibodies to tubulin, antibodies to the membrane. Arrows point at potential contact structures in yellow between the ceramide and the microtubules. The ceramide enriched mitochondrial associated membranes and the microtubules. That's what you're looking at here when you do the storm. When you turn it on, you can see everything's happening in time. You see an event. So the confocal immunofluorescence is all tracked out here. This is intensity marked by distance, looking at this movement along this axis, this line here, in pseudo 3 dimensions, only 2 dimensions. With the mitotracker in blue as the mitochondria, subjected with the immunocytochemistry of antitubulin in red, and the ceramide now in green. Okay, the right panel shows fluorescence signaling intensity and the distribution indicating co-localization of the antigens of the mitochondria. So it definitely looks like our synthetic argument is what matters. You have ceramide, you're linking microtubules with mitochondria, and what that's going to do is induce a serious problem. So there were 5 other figures. I showed you 4. There were 5 others. I showed you 3, I guess. Figure 4 gave you an immunocytochemical analysis, revealed that ceramide co-labeled with contact structures between the microtubule mitochondria, suggesting ceramide is important for function of the membranous contact sites. Using that sphingosine analog, that functional analog, that analog labeled a compartment co-localized a Golgi family protein, and that Golgi family protein is important for microtubule assembly and ER to Golgi trafficking, suggesting that the initial cross-linking of the Pac factor to protein, remember that is the sphingosine analog, was limited to the perinuclear endoplasmic reticulum and or Golgi, with a gradual increase of the labeling over the cell periphery. Okay? Protein cross-linking to that lipid co-localized with ceramide, because you got ceramide to bind, because it can also bind to this lipid, at microtubules, so that you have the ceramide enriched mitochondrial associated membranes all at the cell periphery. Figure 7 showed protein cross-linked to that Pac factor co-localized with ceramide at mitochondria in the cell periphery, which is prevented in sphingomyelin deficient cells. So again, none of that happens. You don't get that nice movement of the mitochondria to the cell periphery if you have ceramide. Ceramide depletion reduced the ceramide-associated tubulin complexes at the mitochondria. This is all just like nailing down that coffin, saying absolutely that synthetic conclusion is correct, right? That you do have ceramide linked with a corruption of ATP released from mitochondria. The ceramide tubulin complex causes this membrane fraction to bind the mitochondria, slows its transport and movement, thus not allowing it to get to the periphery, thus not giving enough ATP to drive this whole system. Because remember, ATP has to leave the cell, right? Because it's a gliotransmitter controlled in synaptogenesis and prevention from degeneration of the neuron. Yeah. And figure 9 shows ceramide depletion decreases complex formation. So get rid of ceramide, you get rid of the complex formation. What complex? Ceramide to tubulin. And VDAC, none of those come together. And cytosolic ATP levels, right? Those are all decreased when this happens. If you deplete ceramide, you get ATP levels in the cytosol back up, okay? And this is shown with inhibiting, having the knockout in the sphingomyelinase pathway or using this chemical mycotoxin to block ceramide synthase. Both times get 40% higher cytosolic ATP, more VDAC activity than the wild type. So the increase in cellular ATP by ceramide depletion from one of two methods was associated with higher mitochondrial motility, they moved around a lot, resistance to the A-beta protein itself, suggesting a novel function of ceramide for mitochondria, regulating ATP levels in the cytosol and eventually being released from the astrocyte as a gliotransmitter. So that includes motility and resistance directly to the A-beta mitotoxicity. Not myco, but mito means toxin to causing that fragmentation of the mitochondria. This completes the discussion. So this is where they put it together. Elevated cytoplasmic tubulin, shown here, that can be induced by stress, right? Lack of aggregation, remember, lack of aggregation of the tubulin into those complexes, those microtubule complexes, or removal from the mitochondrial membrane, from the ER, from the Golgi, from the nuclear membrane envelope, leads to an increased microtubule disassembly, reduced assembly, okay? Led to association of tubulin with ceramide, and the ceramide enriched mitochondrial associated membrane shown there. Okay, so the ceramide associated tubulin wraps around the mitochondria, stopping movement. It's in the stop mode. Now, when the microtubules extend, okay, there's more of it because more tubulin is being used to manufacture them. All this free tubulin is used to make a bigger filamentous core protein for microtubule production. Extend to the periphery, cytosolic free tubulin concentration is gone. No more little red balls here. VDAC is unblocked. You don't block the VDAC. ATP is available to drive mitochondrial transport along the microtubules. You have the go mode, on mode, I would have called it. But they like stop and go. Okay, cool. It's like stoplights. Higher ceramide levels facilitate ATP deprivation. That means it doesn't make it to the cytoplasm. and the stop mode, while low ceramide levels favor ATP release and, of course, the go mode, which is what you want. That's the healthy system. That's the blockage of neurodegeneration. That leads to higher mitochondrial motility in the sphingomyelin deficient or in the fumonisin-treated astrocytes. The rest of this shows you the potential rearrangement of ceramide binding to the alpha-beta tubulins. That's what they put in the tubulin complex. We're trying to show you the composite structure of those proteins, the alpha and the beta regions of the tubulin put together here, associated with the CMAM with ceramide and blocking VDAC. That's showing you this now, this off mode here, the stop mode here, when you've got this entire protein, right? This tubular protein blocking the VDAC as associated with ceramide, because you have ceramide associated tubulin facilitating this process. So this is the schematic of it. This is putting in the protein where they think the contact points might be. That CTT is the C-terminal tail, which they think is plugging up that VDAC so ATP doesn't leave. Okay, so that's the potential, because they say this is like what they think might be happening at the molecular level. You see, you got this membrane there, you got the ceramide-associated tubulin there blocking, stopping up the VDAC, no ATP release. There you go. So we have actually finished here. Once again, this is a picture of Lake Bled in Slovenia, where half of my ancestry is from. And that's me with my wood cutting there. And there's my Facebook. Please go to there to look at all the previous VerEvMed YouTube video lectures, where this one will eventually be planted to. I told you I was going to show you VerEvMed's. There's a website. Go to it. Take a look at what we do. Contact me directly at my email or contact me through the email at the company site. And I'll be glad to discuss things with you, including anything you just saw here. And of course, a VerEvMed, and for you being a potential client for us. So I'm going to stop now, like I always do. Eventually, I'm sorry if this took so long. It was an hour and a half, but we did a lot here. We covered a lot of ground. So I want to just say before I do my sign off, the next thing we're going to cover is the diaeventontology of all the things we covered before. I'm going to put things together in a dialectical argument form. I'm going to look at the events involved, the key events involved, and ultimately what we're going to do is tear it apart ontologically. That is, look at what's really there, right? What's really there from studying the metaphysics of the event as it occurs via dialectical understanding of the process itself. It's process biochemistry, diaeventontological biochemistry, diaeventontological understanding of biological systems, including this pathology that we've been covering, Alzheimer's disease. Okay, so that's the next thing we're going to do. Part of that might end up as a podcast. Part of that might end up as one of these video lectures. Hopefully both. So I'll let you know via my Facebook what's going on. Hopefully this was interesting, stimulating to you even. And right now, all I've got to do is sign off, and that's what I do. And what I'm going to say is, bye for now.